Yes, Mr. Hill.
Dr. Snape, I have just one small topic to ask you questions about. I'm conscious that I haven't taken you to the sections of your witness statement that deal with viral inactivation and heat treatment. You can be assured that the chair and the core participants have access to that statement and will be able to deal with, to go through it and look at it and take it into consideration later in the inquiry. I, however, am just going to take you to the very end of your initial statement because I'm conscious that you chose to end your written evidence in this way, and I want to give you the opportunity of ending this part of your oral evidence as well. It is WITN 3431001, page 110, paragraph 332 of your statement. What you wrote there at the very end of your initial statement of the inquiry is this. Finally, I want to pay tribute to an individual whose enthusiasm, vision and indefatigable commitment made possible a new state-of-the-art BPL. The BPL facility was already substandard in pharmaceutical manufacturing terms when it was built in 1954 as a civil defence project for the preparation of freeze-dried large pool UV-irradiated plasma. Manufacturing on site had been neglected and under-resourced for a further 25 years and was undersupplied with plasma even for the presumed requirements of the late 1970s. From his arrival at BPL in April 1977, Dr. Richard Lane saw what was missing and set about planning the concept, design and build of the B27 manufacturing facility. Dr. Lane's persistence in identifying and securing the plasma needs of B27 to meet demands considered necessary at the time for self-sufficiency and his determination to plan beyond that for the future were key. It is my personal opinion that without Richard Lane's unstinting commitment of time, energy and enthusiasm, B27 would never have been built and the product needs of England and Wales would forever have had to be met by commercial imports. It is deeply unfortunate that, I won't go say why, but that Dr. Lane is unable to present his own views to the inquiry. It falls to me to be a poor substitute. Does that remain your evidence?
Absolutely.
That is...
And thank you.
That is all I have for you, but there are questions from core participants that I will now take you to. And as the chair said, there will be a bit of leaping around from one topic to another. So please bear with me. And if you need a moment to be reorientated as to where you are, then please do ask. I begin with this question. You talked in your evidence about economic fractionation being like a milking stool, and one of the word economic is the one that I've been asked to pick up and ask you about. Does that term economic refer to cost savings or to maximising the scarce resource of plasma, or to both?
For me, the milking stool analogy was thinking about the effective use of the donor's gift of plasma. The fact is that in a real world, if we didn't utilize it fully down the three streams of clotting factors, albumin and immunoglobulin, then the process would not survive and we would not be serving the patients or the donor.
Sully, if we could have on screen, please, WITN 3431001, a separate question. This relates to the difficulties that Dr. Maycock faced in the 1970s in predicting plasma demand. And I've been asked to draw your attention to this section of your witness statement. It's page 112. what you wrote is this: to be trustworthy, self-sufficiency calculations would have required two parameters to be known with a reasonable degree of accuracy. One, the amount of factor VIII required for treatment, as determined by A, the number of haemophiliac patients to be treated and the severity of their factor VIII deficiency; B, the treatment regimen to be followed. 1, treatment in response to bleeding; 2, home therapy, as it developed over time; 3, treatment by prophylaxis, as it developed over time, to normalise life as far as possible; and 4, enhanced treatment in support of major surgery. If we could go on to paragraph 2, please. So that is the first parameter which would be required to be known with a reasonable degree of accuracy. The second is the amount of factor VIII available for treatment, as determined by A, the quantity and type of plasma available for factor VIII production; B, the capacity / capability of the NBTS in England and Wales to meet those demands; C, the capacity / capability of the then unlicensed facility at BPL Elstree to meet those demands. Even with attention to its GMP limitations, PFL in Oxford could only ever operate as a development and GMP pilot scale facility. D, the yield achievable with the manufacturing process, including any confounding effects such as 1, patient-physician demands for desirable product characteristics, presentation, storage requirements, solubility, specific activity, convenience in use; and 2, the impact of any process modifications required to take into account newly emerging risks, in particular blood-borne infectious agents like hepatitis viruses and HIV. So those are, as I read it, some of the complications and variables that were involved in the 1970s and indeed the 1980s in estimating demand. Do you stand by that evidence?
I do, though obviously Sir William Maycock wasn't faced at the time with HIV, but hepatitis viruses were a challenge to us.
And those challenges would have been faced not only by Dr. Maycock, but by others who were seeking to estimate demand in the 1970s and the 1980s as well.
Including the Department of Health and Haemophilia Centre directors and Transfusion Centre directors.
Separate question. You said it would have been extremely difficult for BPL to have assisted the transfusion service in creating a supply of cryoprecipitate as an alternative to concentrates. Why did you say that?
Because at the time in question, in 1983, attempting to, and I make no apologies for use of the term, attempting to shoehorn a new process into an already overcrowded and improperly constructed, inappropriate facility would have done more harm than good.
Following on from that, are you able to summarise the implications for BPL of a reversion to cryoprecipitate in or around 1983, 1984?
First of all, if BPL was to be involved, then we would have tried to assist the transfusion service in turning the clock back, reintroducing some of the issues associated with cryoprecipitate and helping them address those. They've moved on and they no longer had that experience, or they didn't necessarily have that experience in-house. In terms of what we received from the transfusion service, we would have received less plasma, assuming that the plasma was used to make cryoprecipitate at centres. We would simply have had to deal with that, and we would have produced less factor VIII. And we would have had to look at a different starting material, cryoprecipitate supernatant, different cryoprecipitate supernatant produced at 14 different centres to be used to recover other coagulation factors like factor IX and albumin and immunoglobulin. there were considerable difficulties involved.
Am I right in understanding your answer then to mean that it could have been done, but it would have been a very major undertaking?
It would have been a major undertaking. It wouldn't have produced for patients all of the product that they needed. And in my opinion, it wouldn't have produced them the kind of product that they needed.
Why do you say it wouldn't have produced the kind of product that they needed?
Because cryoprecipitate had inherent problems in terms of cryoprecipitate produced as a single donor product was not susceptible to quality control. My experience was that in order to give cryoprecipitate a dose calculated on the basis of 1 unit of cryo was typically administered as 2 units of cryo to be sure to get the amount of factor VIII that the patient needed.
Are you aware of any meeting or series of meetings or discussion papers where BPL, representatives of regional transfusion centres, representatives of the hemophilia centre directors, representatives of the DHSS gathered together to try to come up with, or at least to discuss, the possibility of a coordinated response to AIDS, such as going through the pros and cons of a reversion to cryo?
If such a meeting occurred, I wasn't aware of it and I wasn't present at it.
If there had been a reversion to cryoprecipitate in 1983, 1984, that kind of period, do you think there would have been knock-on effects for the redevelopment of Building 27 at BPL?
Of course, we would have needed a different BPL, if we needed a BPL.
I've been asked to ask if you could summarise the benefits that there were in fractionating through larger pools rather than smaller pools.
I'll try. The benefits clearly are from 1 kilo of plasma, we would recover more factor VIII, and that would make the amount of product available to patients greater. The losses would be largely the process losses of working with smaller batches and the quality control losses involved in taking more product, more vials for testing and making less product available to the patient.
And is it right that there were certain blood products that had to be made through larger pools?
Oh, immunoglobulin, certainly. It was essential. The pharmacopoeial monograph for intramuscular immunoglobulin, which I was part of the committee constructing, specified a minimum pool size of 1,000 donations in order to have the right profile of antibodies present.
Would it have been possible to have continued to make immunoglobulin according to those pool sizes whilst making factor VIII and other coagulation factors in lesser pool sizes?
Yes, by pooling the supernatants from factor VIII to make immunoglobulins, yes, they could have been done like that, and similarly we could have used time-expired plasma. But no, let's stay with the supernatants. That's would have been the practical option.
Is it correct that commercial factor VIII products were perceived to have advantages such as better solubility, higher potency in small volume, lower viscosity, which made for ease of administration for home treatment and prophylaxis?
It was perceived quite properly that that was the case before, when we were still making Blomback fraction in the early 70s. Once we move to the 8IP product, pre-heat treatment, and to the 8CRV product, its extension, and certainly once we moved to 8Y, the FHC product, then I don't believe there was any criticism could be leveled at the BPL product compared with the available commercial products at the time.
You mentioned the Blomback method there and the change to the Johnson method. Are you able to assist in any more detail about when that change took place at BPL in terms of the year in which it took place? I think in your statement you referred to 1974 to 1975 as the point of change for PFL. Can you assist any further as to when it happened at BPL?
I can't give an authoritative statement, but I would have expected to be at the same time.
Do you know why that change didn't take place at either BPL or PFL earlier than 1974 or 1975?
At PFL, we simply did not, I did not have the resource to investigate and establish the alternative to the Johnson product any sooner than we did. Effectively, my work was capitalized on by Dr. Smith when he came to PFL in 1975. Processes of that complexity don't happen overnight. You can't simply import the paper definition for a process and make it work quickly. The freeze dryer, for example, that we had at PFL was geared to freeze drying from bottles. It was not geared to freeze drying from vials, and I'm sure the same was true at BPL.
A further question about process, but perhaps a little later in time. The inquiry heard from Dr. Foster about the continuous small volume mixing process that was employed at the PFC. Firstly, were you aware of that process and what went into it?
I wasn't intimately involved in reviewing it. I know that my colleague David Wesley was, but I also know that that process was targeted at albumin and and worked, at least in PFC's hands, well for albumin. I'm not aware of any evidence that it was extended or scaled up to bigger molecules like immunoglobulin molecules and factor VIII molecules. But that's something I'm sure Dr. Perry may help with.
Do you know if there was any discussion at BPL about adopting a version of CSVM in Building 25 or in Building 27 when it was completed?
Taking Building 27 first, no, I don't believe there was any discussion of the CSVM process being installed in Building 27. I know that David Wesley visited PFC and did an analysis of the CSVM process. His report was not favorable in terms of its appropriate, as being appropriate for BPL.
in terms of building 25, could it have been adapted there and used there, or was the building simply not appropriate for it?
It could have been adopted for albumin manufacture or for the recovery of fraction 5, but our deficiencies in building 25 weren't primarily associated with albumin products. They were the challenge of producing a product like factor 8 in that facility.
Turning to a different topic, which is to do with knowledge of the views of people with haemophilia. I think you said in your evidence that while at Oxford you had some interaction with haemophilia patients, but more with haemophilia clinicians. Is that a fair summary?
Yes.
In your evidence, you said that you gained the impression that physicians treating people with haemophilia considered that the use of factor concentrates was the lesser risk than non-A, non-B hepatitis transmission, that you gained the impression that that was the view of their patients as well. First of all, is it right to say that you never yourself undertook a kind of formal survey of patient views?
I did not undertake such a survey, no.
And so your impression of the views of people with haemophilia is one that is gained from seeing how the products were used and talking to clinicians, rather than taking the views of the people with haemophilia themselves?
True, but not just the physicians at the Oxford Haemophilia Centre, also the other physicians who I met at scientific meetings and at the Haemophilia Centre directors' meetings.
You, as a fractionator, wouldn't have any knowledge of what the patients were being told by their clinicians about the balance of risk. Is that fair?
And that's fair.
Also to do with the end users, as it were, of BPL's product. The size of the pool was placed on the label of BPL product, and you said in your evidence that you were conscious that that label would be seen not just by clinicians but also by patients. What was your understanding of how much use that would be to the patients themselves about the nature of the product?
Yeah, I think that's really inviting a comment, which is for me.
I'll move on from that, Benson. Did you and your team at BPL ever consider doing some form of market research to understand what information patients would find useful in the patient information leaflets or on the label of a bottle itself?
I certainly didn't. I wouldn't have seen it as being appropriate for me to be involved in that. I don't know whether BPL Product Services, as it was called in the early days, or the marketing department as it became in 1990. I don't know if they did, but I certainly did.
On the related question of pool sizes, did you or colleagues at BPL feel any discomfort at the fact that the decisions that BPL were making on the size of the pool involved weighing the risk of greater pool sizes against the risk of greater infection against non-A, non-B hepatitis?
I don't think so, because by the time we were making pools greater than 100 or certainly 500 kilos of plasma, I personally was not convinced that increasing further necessarily increased risk to patients.
On a related theme, given what you've just said about the level of risk involved with fractionation of that size, I've been asked to put to you that the label on the bottle shouldn't have said that there was a possibility that the product contained, the label shouldn't have said cannot be assumed to be free of hepatitis virus. It should have said something along the lines of the user should assume that the contents do contain hepatitis virus. How would you respond to that?
I think all I could say is the phrase was never criticized, the phraseology on our label was never criticized either by physicians or the regulatory agencies or the Department of Health. And it was a carefully constructed phrase. I don't recall seeing anything more aggressive, anything more explicit on any other concentrates made by other organizations.
The chair has the wording on those labels, and I will leave other questions on that for submissions to him. Moving on to a different topic. In relation to the failure to provide research and development facilities at BPL and virology testing and so forth, you said today that Dr. Lane's warnings in that respect were not heeded. Who or which body was responsible for heeding such warnings?
I think it would have had to be upwards from Richard Lane to CBLA, and then via CBLA or NBA, whoever was the managing authority at the time, to Department of Health.
What about the period before CBLA? Dr. Lane took over in 1977 or 78. There was a period of time before CBLA was formed in December 1982. So who would have responded then, would you expect?
I believe that it would have to be the Department of Health. Northwest Thames were played a different role. If Dr. Lane was going to convince or try to convince anyone, it would have been through his links to the Department of Health and to groups like ACVSB, the Advisory Committee on Viral Safety of Blood. Those would be the situation, the locations of power in that sense.
I think probably the ACVSB didn't exist at that time, but there may have been other committees which occupied a role which might have been relevant. I can see that.
Yes.
Different topic. When you said yesterday that some clinicians would have preferred BPL and PFL to promote their services and products more actively, what did you understand to be the aim of such promotion?
I guess the impression that some people received was BPL didn't have the confidence to present its role and its products as positively as we should have done. I can't believe for a minute that those individuals would wanted BPL to take on a very active marketing role, because we were not selling our products, certainly not at that time. But perhaps they saw it as being a deficiency in terms of not presenting a positive front to the people we were supplying our products to.
Different topic, your audits of regional transfusion centres. Was anyone at BPL responsible for going back to a transfusion centre and checking to see if the changes that you had suggested had been made?
It would have been exactly the same individuals doing the follow-up and at the next visit. But there's no reason why one has to physically audit. It's possible to pick up, even then, to pick up a telephone and find out what had been done. And we saw the results in the context of shipments of plasma to BPL.
So there would have been an eye kept on whether or not a particular regional transfusion centre had implemented the types of changes that you had suggested.
And whether the quality of plasma had improved as a result.
Moving on to some of the processes within BPL, you referred yesterday to plasma compromise as being one of the reasons to open up pallets of stored plasma when the inventory hold was in place. What did you mean by plasma compromise, and can you give examples of the type of compromise that you were talking about there?
Yes, it came up again earlier today when on a frequent basis we received notification from a transfusion centre of a donation of blood which had yielded plasma that had been sent to BPL, being involved in post-transfusion, post-cellular transfusion infection in a patient. Then it was necessary for BPL to enter a pallet and recover the specific donation of plasma associated with that recorded infection. That system was core to our quality systems. We had to be able to trace forward from an individual donation of plasma to its pool to the product, and in the event of an event with a BPL product, to trace back from the batch of product to the pool to the donation.
I won't go to it now, but I said earlier that I would flag up parts of your witness statement that deal with some of the processes that were in place, certainly the later 80s, in this respect. And I just put it on the transcript that page 149, paragraphs 96 and 97, and the references contained therein may be helpful to core participants in the chair in that respect. Single plasma packs or international plasma packs. I've been asked to ask why it wasn't possible to introduce those at BPL earlier than they were introduced, which I take to be in the very late 1970s and early 1980s.
The international plasma pack was the specific polyolefin pack that was designed for use with the teardown machine going into the factory. Single donation packs, one of the difficulties, and I wouldn't know all of them because I wasn't involved at BPL at the time, was that there wasn't just one single donation pack. There were a number, and because the 5-litre valet pack was serving us as well as it could, without going back to transfusion centres and saying, okay, we'd like you tomorrow to stop sending 5-litre packs, but send us individual donors, individual donations. That's the only way it could have happened. I don't have enough familiarity with what individual transfusion centres' practices were, or how difficult they would have found it to switch to SPPs. At the time that I started at PFL, all of our plasma was coming from the Oxford Transfusion Centre, and blood was still collected in bottles at that time, not plastic bags.
Is this also another example of how BPL could come up with ideas and could propose ideas and could seek to persuade regional transfusion centres, but they couldn't direct them to switch to single plasma packs?
It would have needed a coordinated activity that included BPL and the transfusion centres. Without that coordination, no, it couldn't have.
A question about the expertise of you and some of your colleagues at BPL. You had grown up through the state fractionation system, as had Dr. Smith, save for the caveat that you gave, and to an extent Dr. Lane as well, again with the caveats that you gave. What steps were taken and when to obtain experience and expertise from those who had grown up through the private pharmaceutical manufacturing system?
When is probably the second half of 1981, when when there was consideration to the appointment of a manufacturing manager, in the case Gilbert Mallory, a head of quality control, me, and an engineer, Ian Ling. Before that, I'm not convinced. I didn't see any evidence that the organisation was looking for people with manufacturing experience. And before that, of course, we're talking about the Lister Institute, and that was not their focus.
Was recruitment of people from the private sector easy, given the salaries involved and indeed given the state of Building 25 at the time?
No, on both counts. We could not afford to recruit from the industry at NHS pay scales. I'll forbear talking about my own situation, but with engineers, we were constrained to recruit on hospital engineers pay scales, and hospital engineers didn't have the experience that we needed at BPL.
Who imposed those constraints on the pay scales?
In the time of the Lister Institute imposed those scales. Subsequently, between 78 and 82, then it would have been through Department of Health and their agents at Northwest Thames, excuse me, Health Authority.
A couple of further questions on pool size, and in particular the later decision to increase the maximum pool size from 10,000 donations to 25,000 donations. What was the motivation for increasing the pool size in that way?
To take advantage of the expected processing capability of Building 27, and again to achieve more vials of factor VIII from a given quantity of plasma.
heat treatment would have been in place by that stage. Did you consider that there was an appreciable increase in risk in expanding the pool size in that way?
By you, I presume you mean BPL?
Yes.
No, we did not.
Why didn't you think there was an appreciable increase in risk?
Because producing a 10,000, producing, say, a 20,000 donor pool in 1 week compared to 2 10,000 donor pools either the same week or the following week was exposing patients to the same very large donation risk.
I've been asked to ask you whether you have a view on what the tipping point was, so to speak, between the pool size, a pool size that would provide some measure of protection against non-A, non-B hepatitis, and the pool size that would not provide a measure of protection against non-A, non-B hepatitis.
On the principle of one donation in 100 being infected, assuming that continued throughout the period of time, I don't think it's possible. Well, I don't think it's possible for me to make that judgment. I'm not I wasn't an epidemiologist. I'm not an epidemiologist, and I would have gone to an expert for guidance.
Discussions about AIDS. You referred in your evidence to discussing AIDS with colleagues, and obviously we have seen the minutes of a meeting at BPL. Did you also have such discussions with clinicians in the 1982, 1983, 1984 period?
Clinicians in the UK, in terms of the hemophilia centre directors meetings and clinicians that I met at scientific meeting, yes, I did. And some of those discussions would also have extended to individuals at the Bureau of Biologics in the States and the Blood Products Committee in the States. So, yes, I had those discussions.
Did you discuss the matter with Dr. Ritzer, who you had a relationship with?
Indeed, yes.
Did the clinicians refer you to the number of their patients who have been found to have HTLV3 antibodies?
I would doubt that the discussions took that line, but increasingly that data was available from publication. So we would have discussed the publications in which such reports were made.
I've been asked to ask about a prefix for a batch, the prefix being HLA, and that being a prefix given to one batch and another batch being labelled LA. Are you able to explain the difference between those two batches? I'm afraid I don't have a document that I can take you to.
No, not without some preparation. I do apologise.
No, please, no apologies necessary. We will think about how we can take that forward in a different way, sir. Hepatitis, and in particular the system for monitoring hepatitis that you told us about, the card system. How did the information come to you about the possible association between batches and transmission of hepatitis?
it would have come from at PFL. The information would have come directly from the Hemophilia Centre and would have been collected by clerks, or my clerk in PFL, recovering the information without patient name from the Hemophilia Centre. At BPL, it would have been reports of post transfusion infection, but they would not have been systematic.
What do you mean by saying they wouldn't have been systematic? There was no formal system in place.
No, if a batch or batches were not recorded at a treatment centre with a record of a patient or patients becoming jaundiced, then we would not have had negative reports in respect of batches. It would have been when there was something to report.
Were you aware of what patients were being told about the use of that information that ultimately came from them and from their condition?
No.
And looking at it from the other end, were you aware of what use Dr. Zuckerman and Dr. Krask put that information to?
Only from the reports of the Hepatitis Working Party to the Hemophilia Centre Directors Meeting, which Richard Lane would typically have attended, or which Dr. Smith or I would also have attended.
So in effect, you would see the published work, as it were, or at least the work disclosed to those meetings, and that was the end result that you saw, but you didn't, other than that, you didn't know the use to which they were putting that information.
No.
What was the reason for waiting until the fourth incident of hepatitis before contacting Dr. Krask?
I can't give you, there was no statistical reason. I mean, there's nothing magic about the number 4, but to begin with, not every batch recorded even one flag. Increasingly, I think it became obvious that more batches were being flagged, presumably reflecting a higher non-A, non-B hepatitis incidence in the donor pool.
Were efforts made to recall a batch when 4 flags were reached?
No, and I don't think it would have been practically possible, because by then, I mean, bear in mind that PFL batches were quite small. By then, the batch would have been exhausted in any case.
Were any steps taken, perhaps after 1 or 2 flags, to seek to restrict the use of that batch? For example, saying don't give it to patients who have not previously been exposed to factor concentrates?
Not that I can remember, but that's not to say that information wasn't being exchanged between the PFL and the Oxford Hemophilia Centre on a discussion basis.
On AIDS, we've looked at the exercise that was undertaken in respect of batch HL3186. Were other efforts made by BPL to try to identify their own products that had caused HIV infection, as in some form of look-back exercise?
I don't know. I don't believe so.
Yield of factor concentrates. You've mentioned that as one of the complicating factors for Dr. Maycock in estimating demand. Was the yield of factor VIII from plasma that was made into cryoprecipitate consistent?
No. If you mean cryoprecipitate made at transfusion centres, no, it was not. But physicians using cryoprecipitate wouldn't have known that, because physicians using cryoprecipitate didn't typically have access to assay facilities that would allow the potency to be determined. I worked quite closely with the Manchester Transfusion Centre with Richard Wensley in a working party studying cryoprecipitate, and it was clear from that working party, which is one of the papers that's referenced in my witness statement, and there was significant variability in cryos, and that was one of the reasons that Dr. Gunson wanted that working party to be assembled and to take place, so that we could provide guidance on how to improve the quality of cryoprecipitates.
Does it follow from your answer that a clinician couldn't be sure how much specific activity there was going to be in a unit of cryoprecipitate? Does it also follow from that if you look back at the plasma that you have with which to work in the first place, you cannot be sure of the type of yield that you are going to get from that block of plasma?
Are you talking throughout all of that? Are you referring to the manufacture of cryoprecipitate?
Yes, I am.
I'm sorry. My head became full of cotton wool.
I think you're being asked, are you about single donor cryoprecipitate?
Yes.
So the case of one single donor is what you're considering?
Working with single donor cryos, the variability in yield, the variability in factor VIII units that could be recovered from it did vary enormously from one centre to another, and within a centre from one donation to another. When you pool plasma to make factor VIII concentrate, you make a pool, you fractionate that pool, and what you're able to do is undertake testing on representative samples of all of the vials recovered from the pool, and then you can give a reliable statement about the yield. For any individual cryo, you could never do that.
Also on yield, you've said that there was a difference of opinion about the yield achieved in Scotland compared to the yield achieved in England. First of all, was that just a difference of opinion, or was there a different yield that was achieved at PFC to BPL? And if there was, what was the cause of that difference?
Sometimes the difference was a matter of statement. A statement that says that plans for a factor VIII yield of 70% simply doesn't bear thinking about. It won't happen. It will never happen. In terms of were there specific differences between PFC and BPL or BPL, PFL? Yes, the most obvious one is the factor VIII assay that we used. BPL, PFL used a two-stage factor VIII assay. PFC used a one-stage assay, and they were. There's lots of evidence of differences according to whether use of a one-stage or two-stage assay. There were also differences according to the standard, the reference preparation that was used. But the difference between BPL, PFL, and PFC, I'm sorry, I would have to say that much of that was down to exaggeration of yield, or exaggeration of yield claim at a certain time by particular individuals.
Can I just ask you about that, the answer you gave in respect of the reference standard? The international unit presumably has an origin rather like the yard in measurement in the UK, or the original pint, whenever that was identified, in some central, as it were, library of the units, and then you can copy that and send round the reference standard. That's my mental picture of it. Is it the same standard in that sort of way, or is it different?
The principle is the same. So every international unit that was used was a concentrate. BPL provided, or PFL rather, provided many batches of concentrate to an IBSC for them to make their international standard. The international standard would then be released in very small quantity from an IBSC, from a WHO laboratory, to a manufacturer who would prepare their working standard. And the working standard, hopefully, was also a concentrate that was representative of the company's products. So had the same characteristic. It was, and that was fine when it was possible to do it like that. But when organizations tried to compare concentrates with plasma standards, which is what was the initial situation in the late 60s, early 70s, then that comparison of a concentrate test sample with a plasma reference preparation is much less reliable. And the differences were not trivial. They could be 20 or 30% differences. And the introduction of new reference preparations on more than one occasion resulted in organizations suddenly seeing their yield, the yield measured in-house, either rising by 20% or occasionally falling by 20%. And I can very clearly remember embarrassing discussions with my chief executive as to why suddenly, because of something happening in an assay, our output of factor VIII appeared to have dropped by 15%. And it wasn't real at all. It was the same process making the same product. It was just the way the assay was seeing it.
Thank you.
Final topic from me. You used the phrase batch size rather than pool size. Could you just explain the difference between those two terms?
For me, the batch size is what, as a fractionator, I begin my process with. It's what goes into a tank. It reflects the characteristics of the equipment that I'm using and the plasma that's available to me. The donor pool size for me only had relevance insofar as we had at any one time a commitment in our process description lodged with the agency of what was our limiting pool size, and that was what was reflected on the label.
Those are all the questions that I have for you, Dr. Snape. I turn to the chair.
Yes, well, I've got 4 questions for you. Can I begin by asking Saleh, if you would, to go to the witness statement, WITN 3431001, and go to page 34. And it's paragraph 94. And it's the last sentence that you have there. The last sentence leads on, as it happens, to the next question I'm going to ask you about. But you're saying the practical reality, though, is that we were under pressure to produce as much factor VIII as possible from a limited supply of plasma and in deficient facilities. And then you describe those. I'll come back to that description in a moment. What or who was putting you under pressure?
Remotely, of course, the pressure was from patient use and physician demand. But locally, the pressure was in terms of a commitment to produce and to increase the amount of factor VIII that we could obtain from the plasma supply available to us. There was, and Mr. Hills referred to it certainly yesterday and I think today, there was a cap imposed on that by the Medicines Inspectorate after the 1979 inspection, quite properly imposed, which we then respected until improvements of the facility, or rather improvements in some local equipment became available under the stopgap program.
Now, these are then pressures in terms of quantity of product. How does that compare with any pressures that there were on you meaning BPL or PFL, depending on the period, in respect of the quality, particularly the safety of the product?
That would always be a balance in the manufacturing process, especially for a manufacturing process designed to produce products for intravenous injection. So at the same time as making the process more efficient in producing more factor VIII, at the time that's referenced in that particular sentence, what we were also trying to do is to compensate for the inadequate facilities within which manufacturing was taking place. So the balance between process throughput and quality was always real, and it was certainly one of the roles of the qualified person to make sure that people didn't forget that balance and that things were really kept properly in balance.
Now, the second question really arises from, hinted at, in a theme to which you've come back a number of times in your evidence, which is the inadequacy of the facilities at BPL throughout the 1970s, as you understand it, and into the early 80s. And if we can just, please, can you leave it up, Sally? Thank you. If we see there, the facilities at BPL, deficient facilities, you say, the facilities at BPL were built in the mid-1950s with little subsequent improvement and few concessions to the developing concepts of good manufacturing practice. That's dealing, I think, with largely with the structure of the building. See paragraphs 235 and one following, limitations of the old facility building 25 at Elstree. Sally, can we go to that?
Page 81.
235, which you'll find at page 81. And you start by saying that the description and limitations of the old facilities at Elstree, including Building 25, the title doesn't really do full justice to the issues involved. Do I take it from that you feel quite strongly about the inadequacy of what BPL was dealt or had to deal with in the early 1970s and following?
Yes.
And you say, 236, the basic infrastructure of, 236, please, Sally, thank you. The basic infrastructure of BPL, inherited from the Lister Institute, was more appropriate to a laboratory engaged in research and relatively small-scale production. And you say how it was built really post-war as a civil defense project for really something which it was not now being used for. And little planning for a fractionation process, the extension to the building in 62, little more than relieve immediate pressure by creating a new bacteriology lab and moving small pool plasma processing into the vacated space. A further extension in 1972 made space for immunoglobulin production and allowed freeze-dried small pool plasma to be replaced by very limited, brackets inadequate, space for albumin and factor VIII production. So, and you go on, really, to describe how it was almost inevitable that the Medicines Inspectorate had the visit they did in 1979 and condemned the building, or condemned the use of it. If it had been a pharmaceutical facility, might not have survived. Do you think that perhaps far too long, or too long, let's start with too long, was taken to renovate or replace the facilities for the purpose that it was going to be used in the 1970s?
I think too long was taken, first of all, deciding that it was not possible to renovate the facility and that the only solution was a new facility. If we had tried, if the solution had been imposed upon us that we had to keep making do and mending in the old BPL, it would have been inevitable that at some point I, for example, as the QP, once I moved to BPL, might have found myself in the position of having to say, this is too much, this is too far. With the decision to build a new facility, then it was possible to see into the future to a building that could deliver safe plasma products. I won't just say factor VIII, but safe plasma products in the quantity that patients deserved. But it wouldn't have happened in Building 25.
So you suggest it took too long. I have been told in this inquiry that in 1967, Dr. Rosemary Biggs was issuing, in effect, what might be described as a clarion call for the increased and improved production of factor VIII concentrates or treatments, looking into the future and seeing that they would need to be manufactured and for the investment necessary to do it. Is it your view that that call should have been answered sooner rather than when it was?
It is, partly because prolonging the use of Building 25 was inappropriate and potentially certainly put output at risk, but potentially put patients at risk, but also because by delaying the decision to specify and build a new facility, we created a situation where importation of commercial factor VIII concentrates was the only resort available to hemophilia treaters. And that then created the situation in which, having experienced concentrate as a way of treating hemophilia, nothing else was going to be enough. Cryoprecipitate wouldn't be enough. The products from BPL were we were not producing enough. And in order to stop that event, we would have had to be beginning the commissioning of a new BPL facility, probably by 1978 at the latest, in order to have that facility producing safe, effective product in adequate quantity by 1982.
The difference between factor VIII, which we've been talking about, treating hemophilia A, and factor IX, treating hemophilia B, could be seen in this way, and I just want to ask you about it. It's the same topic, because the supplies of factor IX produced by BPL and then at PFL were such that very little commercial concentrate was used until there was a problem over heat treatment. Is that right?
Yes.
So
that's correct.
If that had been the case with factor VIII supplies, would you have seen any reason why clinicians, given sufficient supply, would have chosen to use commercial products?
No. The reason, of course, with factor IX is there were fewer Christmas disease patients, factor IX deficient patients, and the demand for factor IX was lower. We were able to meet that demand initially from PFL, as small as it was, and then from the joint efforts of PFL and BPL.
Yes. Well, thank you very much. third topic, in the early 90s, I think, am I right in thinking that BPL operated a shift system in terms of their production?
Yes.
And was that a 2-shift or a 3-shift?
It was both in different departments. Negotiating that shift operation was challenging. Initially, in terms of interactions with union, and then in terms of making sure that we could provide the facilities that operators needed overnight to have food, hot drink. We're not talking about people working in an office. We're talking about people working in cold rooms and working in extremely uncomfortable situations. So yes, we operated a shift system, and it was managed carefully, but it took a while to establish. If I could make a comment there, though, sir, Building 27, the new facility, was designed with multi-shift operation in mind. If someone had said to me, oh, you can put 3 times as much product through Building 25 just by operating shifts, I would have laughed. No, I would have cried, because the single biggest problem with moving from 1 shift to 2 shifts to 3 shifts is the impact of putting more people into a room served with a heating and ventilation, filtered air that's designed for a smaller number of people. In my opinion, had the attempt been made to, for example, at PFC, to introduce 2 or 3 shift finishing activities in the clean rooms fulfilling factor 8 or factor 9 or albumin, that would have failed miserably, because the environmental control tests on the clean rooms would very quickly have disclosed the system was over challenged by particles and microorganisms shed by the number of people, and the system never having 18 hours to rest in between working periods.
So the answer, so far as BPL at Elstree is concerned, is much as you might have wanted to do it in order to increase production, had that been something you wanted to do to meet the pressures that you've described upon you, you couldn't have done it. And so we come back to the inadequacy of the facilities in Building 25.
Yes, exactly.
Which in turn lead to an impossibility or a great difficulty in introducing a revised process which might in itself produce more product.
And a process that at the time, when this was being discussed in 1978, 79, the process that we were going to want to use it for hadn't even been conceived. It was somewhere down the road.
The last point I want to ask you about is minor by comparison, but it's the topic is dealing with identifying the batch which may have gone wrong, might be a rogue batch, might be infected. In the videos which we saw at the beginning, of your evidence, it was plain that a barcode was read for each box that came in. And each individual plasma donation had a barcode on it. When the video itself showed the finished product and described how everything was logged, there is a sight, the only thing I could see was a sight of someone's hand writing in a ledger. Now, what I have to ask you arising out of that is, was the finished product also barcoded?
The batch number was recoverable as by barcode, but I'm trying to picture what you saw in terms of something being written by hand, and I can't think what that was.
I can't help you because I haven't replayed the video to check, but it's my clear memory of it, and it really just gives rise to it's the introduction to a question, which was, how were the individual products recorded? Was it one system? Was it more than one system? I can understand how it might have been written in a book and barcoded, but you can tell me.
The individual vials within a batch were not recognizable by barcode, but the batch number was identifiable by barcode. So it was possible to trace back electronically from the batch number to the individual plasma donations that went into that batch. What you saw in the video when the boxes arrived and the technician was wanding the barcode on the outside of those cardboard boxes, that was an identifier of the box of packs, but within that box were individual donations which were also barcode identified and related to the shipment list from the transfusion center. So it was possible to electronically go from the batch number to every individual donation in that batch by barcode. And if a donation report came in after the batch had been pooled for manufacture, at that point what we have is an index of a batch that's potentially compromised, but we can't do anything other than investigate the nature and severity of the problem and potentially interdict that particular batch.
So in terms of the individual patient receiving a bottle which doesn't have a barcode on it, they will have to trace that back through their own supplier, the hemophilia center, or maybe the hospital, and the hospital or hemophilia center will have to attribute that donation to a particular batch and back then to you, and then you can trace it back. Now that's looking from the patient end back to where it all started, where there may have been an infected donation in the first place, or more than one.
Sadly, certainly at the time that I had direct involvement, we did not have the equivalent of the German blood law, which requires vein-to-vein recording of data. Everything depended, and in my experience fell down on those many occasions I had to investigate it, when you got to the clinic and discovered that the treatment administered that the batch number wasn't necessarily available to you.
Yes, I wondered if that might be the case, and you've just confirmed it. So, well, thank you for that. The next question really is, it's the same topic, the question of identifying when there has been a product failure. A lot of the evidence which we've heard in the inquiry has described how patients with severe hemophilia A had quite a lot of different products. Rarely were they supplied just with NHS, rarely perhaps with just one commercial product. In those cases, it might be quite difficult, I suppose, to trace back the presence of an infection, particularly if the infection happens to be a virus such as non-A, non-B, which wasn't identifiable as a virus by genetic testing until 1989 and thereafter. So you'd have to rely upon clinicians reporting back, and you spoke in answer to Mr. Hill, describing how a clinician may send a report to someone who got jaundice. In the case of someone who's regularly receiving the concentrate, and jaundice which may take 3 or 4 months to develop, were there difficulties in getting any look back as you would see it?
Much more difficult than look back in the case of treatment of patients with single donor cellular products. They can be traced, and the follow-up is relatively simple. But for a hemophiliac who is receiving multiple batches of different products over a period of time, it would be quite difficult and demonstrably difficult to identify which product, which batch might have been responsible for the adverse event that occurred.
Now, of course, I've been asking you questions really from starting at the patient's end with symptoms or an infection and working back to where it might have come from. But it might be much easier, in a way, to trace who might be infected by going in the other direction, from identifying when somebody who's given a donation to a pool has actually come down with an infection or shown signs of being infected. You would rely, I suppose, upon there being reports from a donor's clinician coming through the National Blood Transfusion Service, of which you weren't part, to BPL or PFL to tell you that this had happened, would you?
Yes, and HL3186 was a classic example of what happens when such a report comes in a significant time after the batch has been processed, released, and used to treat patients. You're starting there from an individual donation, which we know went into HL3186, and that batch then went to 2 transfusion centers and was dispatched from there to a number of treatment centers, including Lord Mayor Lower at Alton. And that follow-up then, that's why it was so important to get the feedback from the treating physicians about which patients, and to make sure that samples were made available to John Krask for follow-up.
How often was it that you got a report from the blood transfusion service, the regional transfusion director, who himself or herself would have had a report in from the treating clinician of hepatitis non-A, non-B?
Probably 1 or 2 a month, but usually in time for us to recover the plasma donation from the stock in our cold room. Next worst case, to identify a batch that we'd made from that donation. Ultimate worst case, to identify a batch that had been released and therefore had to be considered for recall.
And so far as non-A, non-B is concerned, that was chronic, and of which there had been very few symptoms during the first 6 months. So nothing much in the acute phase. To see, did you ever have any reports of that?
Well, don't forget that the incidents that I'm talking about are where plasma has been supplied to BPL, but the cellular components from that same donation have been used to treat a sick person in hospital. And they would be, follow-up would be much more effective in that situation, and therefore look back much easier to perform.
Thank you very much. That's all that I have to ask.
Dr. Snape, as with all of our witnesses, we turn to you now and ask if there is anything else that you would like to say at this inquiry.
If I may, please, and it won't take long, but I feel it's important that I say it. It wouldn't have been possible to watch most of the inquiry interviews with the infected and affected as I did, without being moved, often to tears. When I was preparing this witness, or my witness statement, I tried to focus on what BPL, PFL did from 1970 onwards, on what we achieved during that time, and on what stopped us doing more. Much of that was summarised 2 weeks ago by counsel to the inquiry, and I felt with fair and proper focus on 2 key players for BPL, Dr. Richard Lane and Dr. Jim Smith. Now, I know that my evidence is the only oral testimony from BPL, PFL. If I had to summarize what we achieved in just a few words, it would be too little, too late. I know that BPL, PFL staff worked tirelessly, achieved a great deal, but that influences external to BPL, PFL stopped us doing more and doing it sooner. And for that, I'm profoundly sorry. Thank you.
Thank you. I want to thank you in particular. I'm very sorry that we've taken so much of your domestic time away from you. I hope it hasn't been too much of an inconvenience. But it's obvious from the number of questions that Mr. Hill has asked you from core participants, and I have to confess my own time that I've taken of yours to answer my questions, that you have evidence of great value to most of those who are concerned with this inquiry. And it's obvious why that should be. As you said yourself, you're the only live witness that we have giving live evidence about BPL and PFL. So I want to thank you for that, but I want to thank you for more than that. It's quite obvious to me, if I may say so, that you've gone out of your way to try to help in all aspects, not just those that concerned you as quality control manager for much of the time, but more generally, insofar as you could, involving your own research and going back to documents and always with the intention, as I see it, of attempting to help to answer the questions properly and fairly. I want to thank you very much for that. Thank you.
Thank you, sir.
Tomorrow we have Dr. Perry, sir.
Yes. Dr. Perry, tomorrow, 10 o'clock.